Hello, everyone. My name is Andrew Kim, and welcome to the second unit of AP Bio Review Session. Second unit is about cell structures and membrane. Now, I mean, technically, membrane is part of cell structure, but still, membrane is very important, and that is why I separated it in this topic. So, cell structures. Now, first of all, before we talk about cell structures, we have to talk about different types of cells. We have prokaryotic cells, and we have eukaryotic cells. Now, prokaryotic cells are bacteria. Now, bacteria is it's the very element, it's a very basic form of an organism, right? It is the most least evolved organism, I would say. So bacteria, prokaryotic cells will be relatively more simple compared to the eukaryotic cells. Now, when it comes to eukaryotic cells, these are anything that are not bacteria. So that could include humans, animals, plants, and everything, but we will split them into two big categories. First is plant cells, and second is animal cells. Now, technically, it's a bit more complicated than this. We have microorganisms, we have fungi, we have these type of animal, that type of animal. It's more complicated, but for now, for the simplicity's sake, for the sake of getting 5 in AP Bio, let's just remember like this. We have prokaryotic cell, we have eukaryotic cell. Prokaryotic cell is a bacteria, and eukaryotic cell has two different types, which is plant and animals, okay? So now, let's first talk about prokaryotic cells. Why? Because as I mentioned, it's relatively simpler. I mean, bacteria, how complicated could a bacteria be? Now, first of all, bacteria have this capsule, and then they have two layers and something that looks like a tail and a little bit of hair as well. And you have some dots. You have like a weird thread. Now, I'll explain what each of them are. First of all, this tail, it's called a flagellum, and it is literally a tail because when it wants to transport or whatever, they uses this flagellum. Now, what about this hair? This hair is called a pili. Pili is made to increase the surface area. I mean, think about it. Compared to just a smooth capsule, a capsule with furs would have higher surface area because it's more curved, right? So it's meant to increase surface area. Why do you need surface area? Because this, by increasing surface area, bacteria can actually, touch the surface, touch the outer world at a higher area, at a higher surface area, and thus be able to interact with the surrounding better. And interacting can include getting energy or getting whatever components traveling through its body, which is very important, right? So it has some pili, and then we also have a capsule. The outmost layer is called a capsule. The second layer is called cell membrane. Right? Sorry. The second layer is called cell wall. And then the most inner layer is called cell membrane. Now, they're all just similar things, okay? Really, they're just three different layers. It has a lot of layers. Just know that bacteria, prokaryotes have a lot of layers. Now, what's important is what is inside the layer. You have these polka dots, which are ribosomes. Now, ribosomes will appear in later part in eukaryotic cells as well. So please remember what these are. Ribosomes are the parts where they make proteins. Now, protein is not just limited to muscles. Protein could be enzymes. Protein could be synthase. Protein appears in a lot of different parts of any organism, and that is why it is absolutely essential to have ribosomes, which creates protein. Now, this thread-looking shape is, in fact, DNA. This is where DNA is stored in, and it is called nucleoid. I probably spelled that wrong, so just remember remember how it sounds like, okay? Nucleoid. Nucleoid is where the DNA is stored. Now, what's the difference between prokaryotic cell and eukaryotic cell, which I'll explain later, is prokaryotic cell does not have an additional membrane that covers, that sort of surrounds and protects nucleoid. The DNA sort of just floats around in this vacant area inside cell membrane, and this vacant area, by the way, is called cytoplasm, okay? So just remember how DNA is stored is kind of different between prokaryotic cells and eukaryotic cells. In eukaryotic cells, which I'll explain again later, it has additional layer that separates nucleus DNA. It stores DNA in a separate room to protect it. But over here, we don't have that, okay? So that's pretty much it. I mean, pretty simple, right? You have a tail, you have some hair, you have 3 layers, and inside the layer, you have ribosomes, which create proteins, and nucleoid, which stores DNA or genetic information. Also, it has cytoplasm, which is literally just a vacant area in cell membrane, okay? Now let's move on to the eukaryotic cells. Now, in eukaryotic cells, we have 2 different types, plants and animals. I'll explain plant first. Why? Because it's a bit more complicated. So once you understand plant cells, and animal cells will come naturally. First of all, you have a cell wall like this. Inside the cell wall, you have a cell membrane. In that sense, it's kind of similar to prokaryotic cell, right? Except prokaryotic cell had an additional layer called capsule, but plant does not have that. And here you have a nucleus, as I mentioned, and in the nucleus, you have DNA stored. So there's a separate VIP room for DNA. And then you have something called a rough ER, and on rough ER, it has these polka dots, which are ribosomes, and you have smooth ER. You also have something called vesicles. You have ribosomes that are not attached to rough ER. You have something called chloroplast. You have something called mitochondria. You have something called Golgi apparatus, and you have something called a vacuole. Now you might tell me, Andrew, it's sorry, and there is also something called lysosome. Now you might tell me, Andrew, this is so complicated. Why are there so many? Don't worry, I'll break down and explain all of them one by one, okay? Now, first of all, cell wall. Animal cells don't have cell wall, but plant cells do. Why? Because plants don't have bones, so they need additional protection. That's why they have these cell wall that make their structure more firm and stable. You have cell membrane. As I told you, membrane is very important. That's why I'm going to explain about membrane alone separately. So over here, let's just know that there's additional layer. Now, here is nucleus. And as I mentioned, the whole purpose of nucleus is storing DNA, protecting it. Now we have rough and smooth ER, right? Rough ER makes protein. Smooth ER makes lipid or fat. How are you going to remember this? Remember, the reason why rough ER is called rough ER is it's literally the same thing as smooth ER, but it has these ribosomes on it, right? These ribosomes make the surface a bit rough, right? Because it's attached onto it, right? So it makes it rougher, so it's rough ER. And since ribosomes make protein, rough ER makes protein, while smooth ER, which does not have ribosome, cannot make protein, and they make lipid instead. Now, there are some ribosomes that are not attached to rough ER. They also make protein, okay? Now you have something called vesicle. What's important about vesicle is proteins created by ribosomes can't just move in cytoplasm. Again, this same as prokaryotes, eukaryotes have cytoplasm, which is literally just a vacant area in cell membrane, okay? Now, proteins created by ribosomes cannot just travel alone in cytoplasm. It needs additional protected layer, and that layer is vesicle, okay? So when a protein is created from rough ER or ribosome, it moves into this, it's like a bus, right? It moves into this vesicle, and this vesicle will transport the proteins to Golgi apparatus, to vacuole, or even outside of the cell. Now, speaking of that, what is a Golgi apparatus? Golgi apparatus is a place where proteins that are already made are modified. Of course, not when you first make, let's say I make a pasta. It's not gonna be perfect, right? You gotta add some Parmesan cheese, you gotta add this a little bit, that a little bit. So you need to modify before you serve it to your customers, right? Similar to that, once you make the protein, it's not already perfect, so you send it over to Golgi apparatus, where the protein is modified so that it can actually be used outside of the cell. Now, once the protein is modified, it goes back into the vesicle because, again, proteins cannot move by itself in cytoplasm. So this vesicle will once again move the proteins created to vacuole. Now, the reason is vacuole is, as you can tell from how it looks like, is a place where all the waste material and proteins that are created rest. It's like a massive, storage place, right? You store all the waste materials or the proteins that are created already, but not yet can be sent out of the cell. So this is a place where proteins stay for a little bit, and once their proteins can actually exit the cell, once again, vesicles will carry the proteins and deliver it outside of the cell. Okay, so this is the whole process. DNA, or the blueprint of each genetic information of a protein, is stored in DNA inside nucleus. Once the genetic information is copied and moved to either ribosome or rough ER, protein will be created. That protein will transport through vesicle and will move to Golgi apoptosis, where the protein will be finally modified. And once the protein is modified, it's again going to transfer through vesicle to vacuole, where it will rest for a little bit until it can finally be sent out of the cell through, once again, vesicle. Okay, so this is the whole process. This is a whole sort of a mechanism, okay? Mechanism. Now, let me explain what these three things are. Let me first explain this, because it's simpler. Lysosome. Lysosome is like a suicide squad. It's like a suicide button. You click this, and the cell dies. How it works is, as you can see, there's small polka dots in lysosome, right? These polka dots are like dynamites. It just explode and blow up everything. Now, lysosome, when the cell feels like it's unnecessary, it should now just die off. You're already too old. You're not adding anything to the society. Just die. When that moment comes, this lysosome will rip off, all the dynamites will flow out, and it's going to bomb. It's going to blow up, and the whole cell dies off. Now, you might think, wait. That's kind of brutal. Even if you're old, still you got to keep it. You can't just kill the cell, but that is not necessarily true. For example, when you have a lizard, oh my gosh, I hate lizards because they're so gross, but when you have a lizard and I stab him with a knife at its tail, now the lizard is stuck, right? If he can't evacuate, I'm going to step on it and kill him, right? So he got to make a decision whether I'm just going to die here or whether I'm going to cut my tail and evacuate. And that is why around this area, the cells of lizards will break its lysosomes to have all the dynamites blow up and all the cells will rip off. And that is how lizards can actually cut their tail. And without their tail, now it's free. Dobie is free. Lizard is free. Free lizard can now finally evacuate, just run into a corner, run into whatever place is, and not be stepped by me and be dead, right? In some situations, it is necessary to sacrifice some cells, okay? That is when lysosomes are used. Now, let me explain these two things. This is called mitochondria. This is called chloroplast. The reason why I'm explaining these two the last is because they're adopted kids, okay? Adopted kids, they're adopted, so I'm just explaining them the last. Now, why am I explaining them as adopted? It's because, okay, first of all the eukaryotic cell compartments except ribosome have a membrane. I mean, just look at them. You have a membrane over here, you have a membrane over here, you have a membrane over here, you have a membrane. All of them have membrane, right? They have their own layer, except chloroplast and mitochondria. In fact, sorry, not except. Mitochondria and chloroplast also have the layer membrane, but they have double layer. Furthermore, they have their own genetic information. They have their own DNA stored inside them. What this tells us is mitochondria and chloroplast weren't part of a cell initially, long time ago, but through evolution, somehow mitochondria and chloroplast, got merged into plant cell and animal cell, and now they're part of the cell. right? I mean, think about it. Why would they have their own genetic information? Why do they have double layer when only when all the other compartments just have singular layer? These are the hints, these are the proofs that chloroplasts and mitochondria are in fact adopted through evolution. Now, then what are their rules specifically? Well, I'll explain this later in the next unit, but just to tell you, mitochondria does something called cellular respiration, which is creating ATP, which is usable energy. It's like it's making energy so that you can use it. What does chloroplast do? It does photosynthesis. It converts sunlight to sugar so that you can store energy. So think of chloroplast as storage system. It stores energy, and mitochondria is breaking the stored energy to use the energy actually. Okay, so chloroplast is like my dad. It brings energy, it accumulates money. And mitochondria is like my mom. She wastes all the money my dad made. Sorry, that's out of pocket joke. But anyway, you get the point, right? Chloroplast makes the energy, it stores it. Mitochondria breaks the energy made by chloroplast to actually use it, okay? Now, let's move on to the animal cell. Well, there's nothing to really move on because animal cell is literally the same as plant cell, except three things are different. First, there's no cell wall. Why does it not have cell wall? Animals have vertebrates. Well, some of them don't, but we still have bones, right? We have some structure, so we don't necessarily need the cell wall. Second, we don't or have smaller vacuole. Why? Because humans can piss, humans can take a shit, right? We can actually get rid of the waste material on our own ability, whereas plants can't. That is why they needed vacuole to store all the waste material, whereas humans can literally just emit them, right? That is why humans or animal cells would have smaller or no vacuole. Last difference is chloroplast. We don't have chloroplast. I mean, do you photosynthesize when you tan in Cilantro Beach? Do you like what? You grow, you get more energy? No, of course not. Humans eat instead of getting sunlight, right? So humans do not need to do photosynthesis. So animal cells in general would not have chloroplast, okay? So that is pretty much it for the cell structures. And now I'll be explaining about membranes. But before that, let me drink some water. Okay, so membrane. Now, membrane exists in a lot of different places, right? Prokaryotic cells, eukaryotic cells, plant cells, animal cells, all of the cells have membrane, and even some of the cell compartments in plant cells and animal cells had membranes, right? So how do membranes actually work? Now, membrane, first of all, is a layer, right? But more specifically, it's a fluid mosaic model. What this means is fluid. It's not stiff, it's not fixed, but it can kind of shift and move around like almost like a liquid. It's more flexible. Mosaic model means, mosaic, like when you watch, okay, never mind. That's kind of out of pocket. Mosaic, you know what mosaic is, right? Like it looks like this, like these small, rectangular, like blurs, like censorship, right? That's called mosaic, right? Similar to that. You know how when you look at these, it's made out of patches that are connected, right? So mosaic model means it's made out of patches, okay? So fluid mosaic model means if you have a membrane like this, the membrane is made out of a lot of patches of components that are connected that can also kind of flow time to time, okay? Now, that's very abstract, and honestly, this is the first thing we learn about membranes when we learn membrane in AP Bio, but I never really saw this in any questions, so I'm not sure if you have to know this, but just remember, it's a fluid mosaic model, okay? And what fluid mosaic model means is it's made out of patches of different components which can actually kind of flow and have some flexibility. Now, how membranes actually look like is membranes have this layer of something called phospholipid. Now, phospholipid, as you can tell from the name, is a type of a lipid, okay? And they're kind of like, when you go to like a fancy house, you have, or just any house, you have these like gates, right? These fences. Similar to those fences, these phospholipid is just considered as a type of fence, right? Now, what's interesting is if you look at phospholipid, it's made out of two different parts. One is this head, and the second is these tails, right? this head part likes water. In other words, it's hydrophilic. Tail hates water, so it's hydrophobic. That is why heads are sticking outside so they can actually touch water, and tails are just covered inside. It points inside, right? So that it does not have to touch water because it hates water. Okay, so that is phospholipid, and that is how the actual membrane looks like. Okay, now what's so good about membrane is a layer, but it also allows some transportation of components through it. So it's not completely a, a completely separating layer that just 100% separates the inner part and the outer part of the membrane, but it also allows some transportation of components through it. And that transportation happens through two big ways. First is called active transportation. Second is called passive transportation. Active, passive, think about it. Passive people, passive aggressive people does not use energy when they're being aggressive. They're just like, yeah, of course. Yeah, it was great. No, I really appreciate it, right? You're not using a lot of energy, but you're still being aggressive, right? Whereas active aggressive people are, I did not like it. You're being so mean. I hate you. They're using energy, right? So active transport is a transportation using energy. Passive transportation is transportation that does not use energy at all. Passive transport has two different types: simple diffusion and facilitated diffusion, but both of them does not use energy. Okay, by definition, remember this: passive transportation, it's just like passive aggressive people, they do not use energy. If even a little bit of energy is involved, that should be considered as an active transportation. Now, in active transportation, by again using ATP, which is usable energy, components would move, but normally they would move against gradient. In other words, if here's a membrane, and let's say we're moving H plus, okay? H plus. Okay, my computer's lagging a little bit. Now, let's say we have only a little bit of H plus here, but there are a lot of H plus above. Now, I mean, just logically thinking, if it's more crowded, then do you want more people to step into that room? No, you want people to step out of the crowded room and move people to a less crowded room, right? So it's a more natural process that H plus moves downward, but in active transport, we're actively using energy to push people into a more crowded room. So in active transportation, oftentimes components will move against gradient. It will move upward, or not necessarily upward, but it will move from a place where it's lower concentration to a higher concentration. That is active transport. Now, when it comes to passive transport, we're not using energy, so we can't push people, shove people in. But even though we don't actively shove people in, people will voluntarily step out of the room. Why? Because your room is too crowded. So this time in passive transport, it always worked based on aligning, following the gradient, and plus no energy is necessary. Now, then what is simple diffusion? What is facilitated diffusion? Simple diffusion is when components simply just transport through those small gaps between these phospholipids, right? It's just literally trespassing through the membrane. Whereas facilitated diffusion is when the component is a little bit big, it can't simply just move through the membrane, right? The membrane is just going to rip off. So what's happening is there are some proteins, there are some sites where these bigger components can move through so that the membrane does not have to just rip. Okay, so facilitated diffusion is when these components move through proteins, and simple diffusion is when the components move just simply through the membrane. Okay, now then what are the smaller components? Now, what are the components that can move through simple diffusion, and what are the components that should move based on facilitated diffusion? Well, small and non-polar components are the things that can move through simple diffusion. Now, polar or bigger molecules would have to move through facilitated diffusion, okay? Now, here comes a little bit tricky part, okay, which is called osmosis. Now, what is osmosis? How many of you guys have been to jacuzzi or just hot tub, whatever, onsen? Now, whenever you go to those places, you bathe for like say 30 minutes, you're in water for 30 minutes, you walk out, you'll see how your finger is all wrinkly and it's so much bigger than before, right? It's all wrinkly and it's just kind of weird. That is osmosis. Your finger cell has your bodily fluid inside, right? And that bodily fluid, for example, when you cry, that's a bodily fluid, right? When you taste your teardrops, how is it? It's salty, right? It has high concentration. Whereas onsen water, if you taste onsen water, it's just water. Zero, low concentration. When there's a difference in the concentration of a liquid inside and outside of membrane, then there will be a pressure being made. And that is called osmosis or osmotic pressure. Why? Because whenever we have a membrane between two liquid, we want to make the concentration of the two liquid be equal. So if they're different, then we need some pressure that moves water from one liquid to another to make the concentration of the two liquid equal. That is osmotic pressure, okay? And tonicity is basically how strong the osmotic pressure is, okay? It's that simple. Now, there are three different types of solutions. First, isotonic. Second, hypertonic. Third, hypotonic. Isotonic solution is a solution that has the same concentration as the solution that you're comparing to. Now, isotonic, hypertonic, hypotonic aren't definite characteristics. Am I fat? I'm fat compared to my brother, but not compared to, I'm not going to name drop, but people that are fatter than me, right? Being fat is not a definitive characteristic. Similar to that, isotonic solution could be isotonic compared to one solution, but could be hypertonic to another solution, okay? It's a comparative characteristic. Isotonic solution means solution A, the solution you're looking at, has the same concentration as the other solution you're comparing to. So same concentration. Hypertonic concentration, a hypertonic solution is a solution that has higher concentration than the liquid, the solution you're comparing to. Hypotonic solution is a solution with lower concentration than the solution you're comparing to. So if solution A is a hypertonic solution compared to solution B, it means A's concentration is higher than B. That means B's concentration is lower than A, meaning that B would be a hypotonic solution, right? If A is an isotonic solution compared to solution B, it means A and B solutions concentration are the same, meaning that B would also be an isotonic solution compared to solution A, right? So it's a comparative characteristic. Now, one example of isotonic solution is your eye drops. Now, again, when you just pour water in your eye, it itches, right? It hurts. But when you put eye drops in your eye, it feels smooth and very pleasant. Why is that? It's because your eye has some liquid. It has tears, right? Tears are, again, salty. So if you just put water, which is lower concentration, the concentration of water entering your eye and the tear that already exists in your eye won't match. So there will be a difference in the concentration, so your eye hurts. But if you put eye drops, which has the same concentration as your tear, or in other words, is an isotonic solution, your eyes won't hurt because there is no difference in the concentration of liquid that is entering your eye. It's an isotonic solution. What is an example of a hypertonic solution? Well, that could be honey. Now, honey is a very, very high concentration liquid. That is why honey's expiration date is extremely long. You don't even have to put in a fridge. Why? Because honey is high concentration. So whatever microorganism, whatever bacteria, whatever insect that tries to go into honey will die very soon. Why? Because honey's concentration is high, whereas or microorganisms, bacteria, whatever, their bodily concentration will be low. So osmotic pressure will happen and all the liquid inside the bacteria's body will exit its body. So bacteria will lose all of its water, it will be dehydrated and die easily. In other words, no bacteria, no insect, no nothing can survive inside honey because of its high concentration, because honey is a very hypertonic solution. That's one example of a hypertonic solution. Then what's the example of hypotonic solution? As I mentioned, onsen water. When I bathe in onsen water, when I zoom into my finger, it was like this originally, but it's going to look like, oh, like this, all wrinkly, right? This wrinkle happens because all the water goes into your finger. Why? Because your concentration of your body inside will be much higher because it's salty, right? Compared to onsen water. So there will be osmotic pressure happening, which forces water to enter your cells, enter your finger, and make your finger bigger and all wrinkly, almost as if it's trying to expand and just burst, just blow up, right? That's one example of hypotonic solution. Onsen water is hypotonic solution compared to your bodily fluid, okay? Now, that is pretty much it with osmosis. Now, what I want you to now remember is something very important, which is SA to V ratio. Now, SA is surface area and V is volume. Now, some people ask me, Andrew, Rahul Cortez is so tall. Is that because he has taller or bigger cells? No, he has the exact same cell size as us, but he just has more cells. How do you know that? Simple. Because the bigger a cell becomes, the bigger a cell gets, its surface area to volume ratio decreases. So what is surface area to volume ratio, nerd? Then I would say it's literally the ratio between the surface area of the cell and the volume of the cell. In other words, it is how much are you touching your surrounding comparative to your volume. Think about it. When you're eating popcorn chicken versus when you're eating a tender chicken, which chicken, fried chicken, has more crust on it compared to its size? Popcorn chicken, because it's smaller, so the meat is only small, but it's all covered with crust. Whereas tender chicken is big, so in the middle of the tender chicken, there's gonna be no crust, no, that crispy layer, right? So the thing is, SA to V ratio is all about how much that crispy layer you have comparative to your volume. I mean, yes, tender chicken will have more crispy area because it's bigger, but then you're missing out all the middle part with absolutely no crispy layer, making it very soggy and not delicious. And that is why I love popcorn chicken, because popcorn chicken does not have the middle part, or it does have this middle part, but it's much smaller middle part that does not have that crispy layer, right? So now what I'm trying to say over here is, similar to chickens, cells also loses SA to V ratio. It loses this area that touches the surrounding as it gets bigger, because all the part in the middle aren't touching the surrounding. And why is touching the surrounding important? Well, if you touch the surrounding more, it means more interactions can happen between the cell and the surrounding. More energy, more resources, more proteins, more whatever things can move either in or out of the cell, allowing the cell to have a healthier communication, a healthier growth, right? That is why cells should not be so big, because again, if they're big, then they can't effectively communicate with the surrounding. Do we understand? Okay. So that is all about SA to V ratio, and that is pretty much it for this unit, in fact. So that was a relatively short unit, but the next unit is going to be very, very hard, so be ready for that. Thank you so much for watching this video. Great job going through this whole video, and most importantly, don't forget to have a great day.